thought by right you should have completed this. I mean, you should have done that at home. Then let's do a quick recap of where we left off yesterday. We're going to set up the membrane. As we mentioned yesterday, we look at a mitochondria. It's made of two membranes, right? Each membrane has its own name. Okay, assuming this is the cytoplasm. Sorry, I meant you like start. Okay, thank you. Assuming this is the cytoplasm, this is the what space? Okay, intermembrane space. And this is the matrix. Yesterday we touched on the process of glycolysis, right? Okay, glycolysis takes place in the cytoplasm. Okay, so that's the first step. The first step, glycolysis. Okay, let's do a quick recap. Okay, so this is the where. We have where, who, what's involved, what's produced. Right? So what is, who is involved at the very beginning of glycolysis? Sorry, okay, I hear one thing, glucose. Okay, glucose is what's involved. Anything else? Okay. Okay, you need a little bit of ATP to jumpstart the reaction, right? Okay. Anything that is produced? Okay, what are the products of glycolysis? Okay, pyruvate. Anything else? Okay, and ATP. Okay, thank you. And NADH. Okay, so one thing we mentioned yesterday was that at the end of the day, cellular respiration is ultimately an oxidation process. We want to extract out as much electrons from the glucose. So that's essentially what we've been doing. First step, we extract out some electrons, and these electrons will go into NAD+ to form NADH. I'm simplifying this process. The chemists among you will know that something is missing, right? Number one is hydrogen. Okay, so I'm just really simplifying this. All I'm focusing on is that when NAD+ acts as an electron carrier, okay, its role is really to grab all the electrons and then it will change form, become NADH. Okay, so primarily we have a few things we look at products. Pyruvate, ATP, we form NADH. Your homework for yesterday was to look at what happens in the next two phases, right? Okay, so the next phase we look at, if we take this pyruvate and we bring it into the matrix, what happens next? This next phase is called the link reaction. Sometimes in your textbook, they may mention it as pyruvate oxidation. But the idea is the same. When you oxidize something, you're pulling the electrons out. Okay, so let's look at some of your responses from yesterday. So what happens to pyruvate? Okay, let's look at some of the responses. Let's look at Rochian's response. Pyruvate attaches to coenzyme A to form acetyl-CoA, which will be oxidized to form carbon dioxide. Okay, so one of the products identified is acetyl-CoA. Another product identified also carbon dioxide. Let's look at other responses. Okay, let's look at Sanjeev's one. Pyruvate loses a carbon dioxide and an electron during the link reaction. It then attaches to coenzyme A to form acetyl-CoA. Carbon atoms in the acyl groups in acetyl-CoA are completely oxidized to form two molecules of carbon dioxide. Okay, here, from what I'm reading, we seem to understand link reaction as oxidizing pyruvate to form carbon dioxide. Oh, sorry. If we include Krebs cycle, it will eventually be oxidized to carbon dioxide. Okay, so if we were to summarize what the link reaction looks like, your notes, you scroll up, okay, just looking at this step over here, okay, the link reaction. So So students may wonder the where. Where does this step take place? Takes place in the matrix. Okay? It doesn't take place in the intermembrane space or here, it takes place in the matrix. It needs to be brought in first. Okay, if I were to draw the reaction out, this same pyruvate will participate in link reaction. Pyruvate, okay, and as mentioned by Rongxuan and Sanjeev, it will form this thing called acetal CoA. Okay, so that's our next product. Along the way, CO2 may be produced, or rather extracted out. You notice that pyruvate has a total of 3 carbons. But after you extract out CO2, acetal CoA is left with 2 carbons. Okay, this one is just for your information. For your information. Okay, other than that, again we see this common trend. Electrons are extracted out. I'm going to extract out electrons again. This electron goes to NAD+, which is our electron carrier, and we form some more NADH. It seems to us that right now, both steps includes us extracting electrons out. We're doing the same thing over and over. We are oxidizing glucose more and more and more. Remember I told you if we were to extract electrons out all at one shot, it's going to be a lot of energy release. So actually what we're doing here is to extract it out bit by bit. Okay, at every step you find that you extract out a bit more. Okay. Last step. Okay, let's link this last step together. This acetal CoA will then be funneled to the next reaction. Called the Krebs cycle. Good news is that we're not going, I don't need you to know the intermediate steps. Okay? That's for us in SH. Okay, so if you take biology in SH, then we'll dive into the intermediate steps. But right now, let's focus again on the very simple question of where, who is involved, and what are the products formed as a result of Krebs cycle. So if we just simplify this entire thing up, okay, let's say the Krebs cycle is a cycle, right? So you go one round. Okay, what are the products of the Krebs cycle? If we were to just look at this cycle, ignore the intermediate steps, what are the products from it? Sorry? You get 3 NADH, right? Notice that? And when you see NADH, it means that NAD plus has served its role as electron carrier. Again, it's pulling out more electrons. So let me do the same thing here. It is again, once again, pulling out electrons that will feed into NAD plus, our electron carrier, and you get NADH. In other words, the more NADH you have, the more electrons you actually have being stored in it. Sorry, let me rephrase that. The more NADH we see, okay, for every NADH we see, understand that there's an electron being carried. Okay? In this particular reaction, we see that 1, 2, 3 times. 3 times this happens. Do you see a FADH2? Yeah, that's also a product. Okay, so if I go backwards, actually there's another electron carrier that I have not introduced. It is FAD. FAD will form FADH2 when it gains electrons. Okay, so that's another electron carrier I've not introduced in this. Primarily, you see NAD plus a lot. Any other products of Krebs cycle? Sorry? CO2. Okay, you see CO2 being extracted out. Yeah? Okay, so CO2 is another product we find. C is the other way. CO2. Okay, we extract out CO2, specifically 2CO2. Anything else we extract out or form? Anything else that's formed? Okay, GDP and then they bracket ATP, right? Okay, so what it means by that, right, is that the GDP is produced, and this GDP will be used to produce ATP. Okay, so what does this mean for you? You can just write ATP. You can just write ATP. That's the end thing that's going to be produced. Okay, so you do produce ATP as one of the products. Okay, so actually I have a question for you. For every single glucose molecule, how many CO2 will be produced from the Krebs cycle? Okay, maybe we rephrase that. For every glucose molecule, how many CO2 will we generate if we include a link and a Krebs cycle? For every glucose. Okay, 3 or 6? So which one? For every glucose molecule, how many CO2 will we produce at the end of the day?
Oh yeah, it's 6. Because
There are 2 pyruvate, right? Okay, 6 carbons, make sense? Glucose is, after all, C6H12O6. Yeah? So, remember the overall reaction goes something like that. Glucose plus oxygen. And what do you get at the end of the day? You get CO2. Yeah? This is the ultimate oxidation occurring. We have oxidized glucose as far as possible until we get CO2. Other than that, we also get H2O as one of the byproducts. So far, we know where glucose is being used. It's used in glycolysis, right? If I were to ask you specifically where is CO2 produced, you know exactly where to, right? So if someone were to ask you, where does CO2 come from your body? Well, actually now you know many levels deeper. Other than the fact that you breathe out CO2, if we go to the cells, actually CO2 is produced at the link reaction as well as the Krebs cycle. But notice we haven't quite answered a few questions. When is O2 actually used? So far O2 has not been used in any step, right? Yeah, that last step, so we are waiting for that step. When is O2 used? When is H2O produced? So far every step here we don't see that happen yet. Right? So we are saving up for the big game. Okay, so broadly, 1, 2, 3, before we go into the final game for today. What's the commonality between 1, 2, and 3? See any patterns in the interactions? There are electrons being taken out, right? And every step of the way, we oxidize glucose a little bit more, a little bit more, a little bit more, right? Remember, after all we are doing right now is oxidize it step by step. So if you want to take note, right, this is the thing you want to take note of. In every single step, we oxidize glucose a little bit more. We pull out a little bit more electrons in every single step. You might be wondering, whatever for, right? Okay, they are after all called electron carriers. Their role is going to come in really important in today's lesson. Okay, I want you to pay attention today. Where do we use all this NADH? We have been saving this up for today. So you pay attention to this today. Where are we going to use this? Okay, I'm going to take a pause for you to touch up your notes. If you have any questions, now is a good time to also ask and clarify before we move on to the final part of today's chapter. Okay, so take this time to buzz with your friend or raise any questions that you may have. Yeah? Okay, where's Debbie? Okay. Okay, so I want to reassure you, I'll never be asking you to reproduce this entire process. Okay? What's important for you when you look at this chapter is to plot the lengths of interactions. Where does interaction take place? Who is involved? What is the change? Rather, what is produced? You can understand it from this lens, then split it up. Okay? So some students find it very daunting when they see this, like, oh, just highlight the ones that are key to you, yeah? Okay, today is the highlight of how our body produces ATP. It's such a fascinating process, how our body produces ATP. More specifically, our mitochondria. Okay, if you are ready, you can go to the tab C. You go to, folder B. This is where we are going to be today. Folder B, oxidative phosphorylation. It's the very last part of cellular respiration. Okay, before we watch this video, let's look at it from the lens of interactions again. What is needed for this very last step? Okay? Yeah, so we keep this question in mind. What is needed for us to carry out this last step? Next question. The things that we need, where do they come from? Okay? So we bear these two questions in mind. Now, we're going to watch a quick video just to give you an overview of how this very last step works. I'm going to show you this video first because at first glance, if you were to read the paragraph in your textbook, it's mind-blowingly complicated. But if you watch the video first, right, you realize actually it's quite an elegant process. You see how it works, right, the words make more sense. Okay? Perhaps, therefore, I'd like you instead, will you just read this paragraph first? Okay? Read these 3 paragraphs, then we'll see how it moves, how everything comes together, and then after that, we'll do a little bit of role playing. Questions or queries, you might want to pen it down for them to be answered in a moment. For those that are reading, I invite you to look up. Yeah, I know. That done, you visualize how this will flow. Okay, what's the game you're done reading? Will you look up? So you're done. Okay. Alright, now I'm going to screen the video. Okay, so mentally as you're reading, could you visualize what's happening? Okay, so now let's see if the video complements what you just read. Then our final step is we're going to do a little bit of role playing. Okay, so later on, we're going to go out of class. Okay, I've cut out and create small little necklaces. Okay, it's not very small, it's quite big. Okay, for all of us here today. Later on, we'll get into our groups and we're going to try to model and role play this process. Okay? I want you to feel the process. To really understand how it works, you've got to be the mitochondria. Or some of you cringing already immediately. Later you do it with me. Okay? So everyone, later on, you'll get as much as head. You'll be the inner membrane. Okay? So as you watch this, I want you to try to understand how it works. Because later on, we're going to role play. We're going to pretend like, we're going to give the best skit ever to represent how this process works. Okay, right now I'm one of the enzymes we define them. You come across the enzyme Atp Synthase? Okay. Yeah, so let's watch the video. Because later on we're going to go out and we're going to have some fun.
Our body is made up of trillions of cells. They all require energy to function. This energy is created within our cells, in the mitochondria. Here, food is converted into chemical energy called ATP. ATP is released by the mitochondria so cells can use it. Mitochondria consist of two membranes, an outer membrane separating it from the cytosol, and an inner membrane surrounding the so-called matrix. The area between these membranes is called the intermembrane space. ATP is generated at the inner membrane of mitochondria by an efficient mechanism called oxidative phosphorylation, involving several membrane protein complexes. Nutrients provide high-energy electrons in the form of NADH, which are used by the protein complexes to pump protons from the matrix to the intermembrane space. This continuous pumping creates a proton gradient, where the positively charged protons are attracted to the more negative matrix. When the protons re-enter the matrix through the Atp synthase protein complex, they catalyze the production of ATP.
Okay, got a rough idea of how it works? Yeah, okay, so this is where we are going to be. Right now, imagine we zoom in. We are zooming into the inner membrane. Okay? At the inner membrane, you find lots and lots of proteins. And these proteins are who you are going to be today. At the same time, there are other parties involved, okay, that we're going to role play later on. This is what's over here. Okay, so everyone will take up a row of protein later on. You'll find inside this Ziploc bag a whole lot of large chains. Okay, large necklaces. On top of that, inside, you also find a lot of other things. Like, you'll find some hydrogen protons. Okay? If you read your textbook, you'll find them useful in a while. You also find things like FADH. Remember, we created a lot of FADH, right? You also find things like NADH inside. Okay? So, now, can I tell you to bring your iPad along? Okay, we're going to go out of class to do a little bit of activity. So cold. Okay, you can wear a jacket. Okay, so can you bring your iPad along? We're going to go out of class for a while. You just follow me, we're going to find an open space where we can dance a little bit.